again, everybody. We will now start with the first afternoon session, so to speak. The title was already given by Giovanni, Plant Genetic Resources Information Systems, Making Data Fair and Useful. And the objective is to highlight the significance of the FAIR principles and to craft synergistic strategies towards the standardization of the management of plant genetic resources data. Speaker will now be Clara Pons, and she will talk about towards a harmonized system of standards for PGR phenotypic, image, and genetic data. And she will give her presentation online.
Okay. Thanks, Stephan. I'm Clara Pons from the IBMCP, and I'm going to present a talk about harmonization system and standards for PGR phenotypic, image, and genetic data. As Catherine has explained, EURISCO is a central entry point for a section of specific information, passport, and phenotypic information of PGR maintained in Europe. Currently, each country national inventory submit passport data using the multi-crop passport descriptor, but in the case of phenotyping data, only a few descriptors are used, and they don't follow any standard and not fulfill fair competence. EURISCO also don't have information about images and genetic data, but the provision is to extend or integrate this kind of data into the EURISCO system. However, there are no agreed standards to link image and genetic data into EURISCO. To integrate image and genetic data and also to make fair the phenotyping data, it's necessary to employ minimum reporting yieldedness with semantic standard that accurately define metadata using common languages, use unique identifiers that unambiguously and stable identify the objects, use open source standard formats and communication protocols between databases. There are two models to verify data. One is to create new standards, and the other is reuse the existing ones, adapt them if necessary, and promote the use. We have chosen this last approximation, and in this talk I will review the current standards regarding for phenotyping, image, and genetic data, making interest in the strengths and the weaknesses. Regarding to phenotyping information, the MyAPI, the minimum information about plant phenotyping experiments, is one of the most recognized standards for documentation and exchange plant phenotyping experiments. It is being adopted as the MY standard for describing plant phenotyping metadata in several initiatives such as Transplant Project, European Plant Phenotyping Network, Elixir, and Emphasis. All of these projects or initiatives are promoting the adoption. In addition, there are some tools, databases, or projects that have adopted it as a main standard for describing plant phenotyping data, like the FIS, the GMPs, the PIPA, the COPO, or Germinate that now is in the process of integrating, adopting this standard. MyAPI is designed to handle a data set for crops and woody plants grown in different environments, in different years, and actually provides metadata checklist for 83 descriptor anthologies and a metadata model to adequately describe plant phenotyping experiments. These descriptors are included in 4 main categories: investigation, and that includes information about the title, the objective of the description, the author list, citations or publications, the study that describes how the experiments are done, the location, the years, the coordinates, the environmental parameters that are used, or the conditions that are studied, or some events that are occurring during the experiment, and the description of the experimental design and the observation units, how the observations are taken, the biological material info, the source, the taxonomy, the multi-crop passport descriptor information, or if they are harvesting samples to do the experiment, and the observer variable, how a measurement is made, that usually is composed by a trait, a method, and a scale, and the data file, that it's a file, the file itself, or a link for a holding repository where this experiment is. To provide definitions for unambiguously identify a scientific term, there are a lot of ontologies. The most common for plant phenotyping experiment are experimental factor ontology, phenotype and fact ontology, plant ontology, trait ontology, and so on. This allows to define well the experimental conditions, but also the traits or the observation. In some cases, these ontologies do not cover all the traits that are analyzed in different experiments, but it is possible to require a new ontology. These ontologies that I put here are generalist, but most of ontologies are designed for organism or species specific. Like the crop ontologies that you see here, the crop ontology is 302, or the tomato ontology or solanacea ontology. But for example, for the less economical species, we don't have ontologies. So in the case of Jaurisco, we need to develop at least some ontologies, but for this kind of species. To ensure consistency and interoperability, it is necessary use some models to interchange this data. For MyAPI, actually there are 3 models: ISATAP, BRAPI, and biosamples. Each one has the pros and contras. The ISATAP is a structured format for organizing and describing experimental metadata, and data allows interoperability. It's widely adopted, and it's a general solution for organizing experimental metadata in various research contexts. In addition, there is a tool to interconvert MyAPI to ISATAP. The contrast is that set up this kind of table is high time consuming and needs lots of data. In the case of BRAPI, it's a web service implementation that includes MyAPI, multi-crops, passport descriptors, and also genotyping data. It allows to interchange and access data between different software. Is very popular in plant breeding and agriculture, and also there includes a tool to convert BRAPI to MyAPI. So the contrast is that is primarily focused on plant breeding and needs expertise to use and implement. The last one, the JSON metadata standard for biosamples, ensures that the MyAPI interoperability with the ENA submission process to the European Nucleotide Archive submission process. There's also a tool for convert MyAPI to biosamples JSON, and the problem is that it's only specific for ENA. The choice of one of them, of course, will depend on the system that we are using, or the system that we want to interact. Of course, if we want to interact with ENA, we will use the JSON ENA, but for other tools or retrieve data for the Jaurisco, we will manage one of the other. In the case of image standards, actually there are no metadata standards for collecting and displaying plant genetic resource images. We have proposed a series of minimum metadata required for collecting images, that they include general information with the image ID, the institute, or some passport information about the accession that we take the images, some plant parts, some information with ontologies for the description of the specimen, the part plan, the developmental stage, and so on. But also some technical information about the image acquisition, such as the resolution, the device that we have used, the light conditions, the color calibration, or if we have done any process images. And later, some additional notes about the use and the rights and the usage restriction of this image. There are several standards that can meet with the needs that we, for these minimal standards. So for general information, we have multi-core passport descriptor, Darwin core, Dublin core, Outcome core, the ISAN framework, the access to biological collection data, or BRAPI specification. For the part of experimental context where the image was taken, or the plant part, we have MyAPI, the ISA, the BRAPI specification, Outcome core, Dublin core, or ABCD. Or some ontologies, like the experimental factor, plant ontology. So all the ontologies that we can use in MyAPI experiment, MyAPI template. Regarding to image technical metadata and processing, there are some specific standards for image, like the exchange image file format, data dictionary with the NISO standard, or the Dublin core or Outcome core. Now, at this moment, there are no information, so we need to, if a metadata standard for image, so we need to develop one or adapt these ones. In the case of genetic data, there are lots of standards for collecting and displaying data. In a survey in the file sharing page, that is a catalog, a curated catalog of data metadata standards, we found that there are 873 knowledge bases and public repositories related to genetic data, and 297 standards that include identifier schemata, meaning reporting guidelines, models or syntax, and for metadata and semantic standards. Of them, 32% can handle with data and metadata across all species for a particular domain of area of studies. This is genome, molecule, or variations and others, and the remaining 67% were species-specific, tax-specific, or family-specific for the analysis for the standards to be used in the case that we implement genetic data into this. So we exclude this kind of standards because they don't allow to use a lot of species, and as Catherine says, has told before, EURISCO contains lots of species, thousands of species. In the case of identifiers.org, that is a collection for identifiers that are the unique identifiers that has every databases and public repositories, we found that there are 350 IDs related to genetic data. So when we analyze this 32.5%, we found that there are 440 genetic databases, public repositories, and standards accepting a wide range of species. Most of them are provided by the International Nucleotide Sequence Database Collaboration, that includes the NCBI in USA, the EBI in Europe, and collaborators, some like Australia, and the DNA repository for Japan. These databases accommodate every type of genetic data, from raw sequences to curated data, variants, and some tools to analyze or visualize them. They operate independently, they synchronize the data periodically and apply similar quality standards and metadata, all of them. In addition, they have permanent funding. Of course, there are other databases, and some of them are specific for a published paper and are small and no longer maintained, or are specialists, quite important in the field they are created, they are maintained, but usually they send all this information, or this information is stored in the ENCPC repositories. Regarding to standard data files to store genetic data and also to exchange, there are as many as type of genetic data. Here in this table, you can see the most common and widely implemented standard in genetics. They are categorized in 4 categories: sequence formats, alignment formats, variant formats, and discrete genomic feature data, or data tracks for visual representation. All these kind of files have a common structure, that is, they have a head starting with a symbol with some kind of metadata or more, and a body of structured and standardized genetic information, such as the sequence, the alignment, the variant files with the genotyping. In the head, some of them has, or most of them has quality and information, metadata information about the quality or of the sequence or the filters applied and the method. However, there are not. Wait a minute. Okay, regarding to PGR metadata, most of them do not have information. The only ones that have some, and if they have some information, is not fully completely compliant with MyAPI or multi-crop passport descriptor. For example, the NCB, the gene bank or MBL or DGB format, the flat format that stores nucleotide or protein sequence, contains information about organism, the strain or the variety, the taxonomy, author, and publications. For the VCF, actually, it doesn't have any information in metadata, but the past years, a series of recommendations for IDs and URLs to link to every repositories, this information was published. And the last one are the GTRACs, the GTRACs format that are used to represent intervals and additional information for G browsers. They are recently created, and they contains information. They are the most fair standards. They include investigation, study, sample, and experimental and analysis information in an ISA tab format. So we need to find a solution to make this MyAPI and multi-crop passport descriptor correct. And at this moment, for the sequence or sequencing or experiments, the most accepted standard for genetic data are the family of the minimum information about any sequence. They are adopted as the main standard for genetic metadata in the ENCDC collaboration or the Genomics Online database. These families provides a checklist with more than 100 descriptors, ontologies, and environmental packages to adequately describe genetic experiments. This data is organized in 4 modules: investigation, nucleic acid sequencers, sequencing, and environment. Investigation, relate the project and link it to other electronic resources, such as the MyAPI, for example. The nucleic acid sequencers is how DNA, how the sample was created, and the libraries that you have used, sequencing, information about the sequencing instrument, and how it was processed. And environment is the environmental conditions that this DNA sample was obtained. The mix family is divided in several checklists and extensions. For a specific type of sequence, there are for the mix that are for genome, the MIMs for metagenomes, and the MIM marks for markers. In addition, for genomic sequence, there are also specific sub-checklists for different taxa groups: E for eukaryotics, B for bacteria, PL for plasmid, virus, organelles, and plant. This last was created a few years ago. In the case of MIM marks, we have also the survey and specific specimen markers. One of the weaknesses of the mixed standards is that sample and investigation information are not fully compliant with MyAPI and multi-crop passport descriptors. One important information that we have with genetic resource is data, linking data with phenotype with genotype data. This generates a lot of data, but however, the standards and public database for phenotype variants associated metadata are developed for human medical biomedical research. In plants, there are some initiatives to standardize QTL and GWAS or to store QTL and GWAS variants, but usually they are private or organism specific. Only a few initiatives aims to involve a large number of species, the MICAs, the GenPs, and the Elixir plant genomics tool assembly. The MICAs is a set of rules of checklist accompanied with written in XML and ISA formats that describes experiments, population, traits, markers, maps, genotypic and phenotyping results with well-defined statistics. However, it's not multi-crop passport descriptor and MyAPI compliant, and despite the huge effort done for do this standard, is not currently maintained, not widely implemented. The GenPs template is a complete and aggregate fair compliant templates and ontologies for QTL, meta-QTL studies, and GWAS. This data is a very complete set of templates that can be used as a starting point to create new standard for QTL and GWAS. However, it needs to improve for the passport metadata for the panels using the GWAS. And the plant genomics tool assembly are as a set of instructions for submitting plant genomics and genotyping data to the European Nucleotide Archive, or the EVA, the European Variation Archive, ensure that phenotypic and genetic data interoperability through biosamples, that is another repository for samples in the EVI, and also this allows visualization and reuse in assemble plants, Plaza, or further. Harmonizing all of this genetic data needs for ontologies. Currently, there are two ontologies that are strictly used for genetic annotation, like the Go ontology and sequence ontology, and the other ontologies that we need to describe metadata are described previously for the MyAPI, that includes all for ontologies to describe how samples and protocols are obtained, and the concepts and relationships related to phenotype and experiments. As Catherine explained before, the use of sharing the identifiers is essential to link genetic data to a scope. In file sharing, there are more than 300 IDs for genetic data, and when we look at that, we saw that with the exception of centralized repository like the EVI and CBI and others, most of the identifiers are related to species-specific, taxa, or family-specific. In the case of ENSDC IDs, the sequence accessions number have a specific formats. Here you can see the EVI IDs for genetic metadata. They vary from the Japan repository or the NCBI, but they generally follow a similar pattern and have direct correspondence between databases. One of the main problems that we observe with the IDs is that their genetic data usually do not have unique IDs. So we can find the same gene or the same sequence in different databases, normally with different IDs. This at the end produce that we can find the information, or we lose information about some sequence. There are two strategies. One is use persistent unique identifiers, such as the digital object or digital genetic object, or the other is the strategy that the assembler have used. Assemble plant has decided that if a species or organism is well sequenced and there is a large community of people that is working with this organism, they use the organism-specific ID because this is more familiar for the people that is working this, for the researchers that are working this. If not, they use their own identifier. We thought before, the models and syntax standards to storage and exchange data are very important. For genetic data, we can find the XLL, the RDF, the JSON, the shadow, the ISATAF, the OVL, or the BRAPI. For example, the XML, the JSON, and BRAPI can exchange most of type of data, of genetic data. The RDF and the OVL are more related with ontologies, and the ISATAF for metadata related to genetic experiments. The choice of one of these, which is the best, so it depends on which database, which systems we want to interact. One thing that we have observed in our review of all of these genetic standards is that there are no quality standards for genetic data. So when you see also for the databases, the responsibility to submit high quality data lies on the authors. There is no quality standard for submitting sequence, and so we need to establish QCL threshold standards to submit or integrate PGR genetic data in EURISCO. We have reviewed some consideration to ensure high quality data, genetic data, to integrate in EURISCO and to submit to public repositories, such as sequence quality, that there is a good base call, that the WITRIME sequence and adapters, that the alignment quality or filtering on poorly aligned sequence was done, the variant calling, that exclude low quality or variants with low depth, and with some allele thresholds, remove duplicates, analyze the coverage, or set up a coverage depth for coverage, some contaminants, batch effects, replication, genotype concordance, and variant annotation. So one suggestion is that we need to set up some thresholds to say, okay, this is a good sequence, we can set this, we have trust that this data can discriminate well between accessions. Just there is some slides to finish. One of the conclusions that we have arrived after this review is that we don't want to invent the wheel. So the best is to use repositories, knowledge bases, and standards implemented in the international ENSDC repositories, especially those hosted the MBL-EBI, like we are in Europe. And this will provide some benefits, that is, we will do minimal changes, maximize standardization, ensure long-term storage and interoperability, and also funding. That is very important. To do that, we need use serial identifiers or accession numbers to link genetic data between ENDC or EBI with the corresponding metadata in EURISCO. Use common standard, and we don't have this common standard, convert the data to the standards that one system uses. Develop integration methods and APIs to share and exchange, to facilitate data exchange between both systems, and also consult experts and foster universal adopter doing courses, tutorial guides, creating tools, but also involve editors and publishers that press scientists to do that. Just to finish, as a summary of all the gaps or recommendations that we think that are necessary to implement standards and integrate image, phenotyping, and genetic data into EURISCO, is adopt the Miami standard. And if it is necessary, select the most suitable descriptors to be implemented in EURISCO. For image, reach a consensus on adopting a specific image standard or adapting them to meet the specific requirements for plant genetic resource by incorporating multi-crop passport descriptor and MyAPI metadata, along with plant-specific ontologies. For ontologies, develop and adopt agreed-upon ontologies for annotating plant phenotyping, image, and genetic data, especially for less economical important species. In the case of genetic data, use the Elixir Plant Genomics tool assembly for genetic data submission to the EBI. Use the establishing standard file formats, and if possible, enrich with PGR metadata, especially for sample and accession ID and links. Use GTRAC files with rich multi-crop passport descriptor and MyAPI metadata. Redefine mixed standards to be fully compliant with multi-crop passport descriptor and MyAPI. Develop or adopt agreed-upon QTL and GWAS standard for the plant community, and create a repository or promote the extension of EVA, and define quality control transport standards for each type of sequence submitted to the EBI repositories. In the case of identifiers, use the same identifiers between EBISCO and the EBI for study, sample, GenPlus, genome, and to ensure interoperability.
Many thanks, Clara, for this very comprehensive overview. Again, also for your presentation, we will have the discussions at the end of the session. We will now continue with Joana, who will talk about data standards for in situ PGR conservation.
Okay, thank you. So in this presentation, I'll talk a little bit. It's a short presentation because there are some points for discussion that I really would like to tackle at the end of the presentation, maybe in the discussion session. So I will talk about the context of this and the aim of this presentation, challenges for documenting in situ populations, then the existing data standards and descriptors for in situ populations, interfacing with EURISCO, and finally the points that I would like to discuss. So in this slide, you can see this continuum that goes through diversity, existing diversity, conservation, and utilization. And at each step, at each point of this continuum, there is data produced and data needed to go to the next step. So for wild and cultivated PGR diversity, we do need we have basic passport data like coordinates, species names, etc. You have detailed population data such as number of individuals, area of occupancy, etc. Then if you go through this existing diversity until the conservation, ex situ and in situ management of this diversity, then you then have more detailed population management data and monitoring data, say the change of number of individuals throughout time, management regimes, etc. And then you have access to this data, which is essential to be able to utilize this diversity. And of course, then we have this interfacing with EURISCO, which is essential to provide access to this data. So what I thought of this session would be interesting is to get some feedback from you regarding the development of this system. So thinking about challenges in documenting in situ populations, several people here already touched some of these challenges, and indeed you have several issues. Perhaps not all here, but these are the ones that we thought of. So first of all, we have PGR diversity, wild PGR diversity, cultivated PGR diversity. These are different elements of diversity with different requirements. Cultivated diversity versus wild. Then within the wild diversity or the wild PGR diversity, you have crop wild relatives, you have wild foods, and also other elements of diversity. And each of these groups also have specific requirements. Then there's a great diversity of data types. You have basic passport data, as I mentioned before, species name, location, coordinates, etc. The detailed population data, number of individuals, threats to specific populations. Then you have data associated to land races, to the farmers that cultivate those land races, and the specific farm where that landrace is cultivated. Then you have for wild food plants, you have all the ethnobotanical data associated, so uses of those specific populations, the parts of the plants that are used, etc. Then when managing in situ population, you also have a great diversity of data, monitoring data, as I said, and detailed in situ conservation or management data. So each of these types of data includes several factors that we need to take into consideration. And then we have the issue, who manages this data? And actually, how we do it. We know that nature conservation organizations usually, they have the mandate generally to conserve wild diversity. But what about the gene banks? And what about the genetic resources center that Nigel also mentioned? So are these two different elements that, or these different types of institutions that we don't know who is going to actually manage this data? And then accessing in situ populations. Should we access directly from the wild? Should this go via gene banks or genetic resources center? I think Nigel will talk a little bit about this afterwards. So regarding the existing data standards, there are some information, there are some data already out there. For describing populations, in situ populations, we have the principles for the inclusion of crop relative data into EURISCO, which was developed by Theo and José Iriondo from Spain. And this is a very good starting point. Then for the detailed population management data, we in Birmingham and with several colleagues throughout the years, we have developed several descriptors that haven't been finalized, but we hope to do it soon. But then for the monitoring and for the detailed management data, we don't know anything out there. And as perhaps you know more than us, and that would be great to have your input on that. And then accessing the data, again, we can use this publication by Theo and José Iriondo. So just a brief history about the crop-wide relative standards and descriptors. Since 2002, at least, they have been developed intensively throughout several projects in the PGR Forum project and the E-Group project as well. Then we had the publication by Imke Thurman, the core descriptors for in situ conservation of crop-wide relatives, published in 2013, also a very important publication. Then we had several descriptors and tools developed in PGR Secure and also in a SADC crop-wide relative project, which took place in Southern Africa. Then also Stephan Weise, also here, and colleagues developed this concept for possible extension of EURISCO for in situ crop-wide relative and on farmland race data. Very, very important as well. The FAO released the FAO descriptors for crop-wide relatives conserved in situ very recently, and then again Theo's publication, José Iriondo's publication on accessing crop-wide relative data in EURISCO. Here. Now, for wild food plants, I must say I'm not so knowledgeable about. So. Our idea was to adapt what exists for crop-wide relatives, but obviously this is not ideal. So I would like to hear from you, maybe afterwards, if anyone knows about these descriptors that actually describes populations of wild food plants in situ. But they are wild food. They're wild plants like crop-wide relatives. So perhaps the existing descriptors, if there are none already developed, we could adapt them to this other group. And then for land races, for several years now, it has been a lot of work has been done as well. So basic passport data, we have this the publication by Stephan already there. For detailed population data, publication by Valeria Negri and colleagues. And then for in situ population management of landrace is also publication from 2020. It's there with some descriptors and monitoring data as well from Valeria's publication. And then accessing in situ populations, again, Stephan's Weise's concepts on land races and crop relatives. And here are perhaps the two most important for this particular issue, for this particular project, these two publications. So again, accessing, I must confess that for me, the basic population data and the data for accessing these populations was a concept, a difficult concept, because for me it was the same. So you have basic population about an in situ data, which is probably the exact same thing as the data that you need to provide for those who want to access those data. So I'm just thinking out loud. But basically, we already have these descriptors, so it's I think it's we are in a good shape to go forward. Now, interfacing with your RISCO, this is something that we want to discuss with you. This concept was, I think, is the same as for ex situ. So basically, there's sites that conserve in situ the crop pod relative and landrace population, and then there's a national focal point that gathers this information and then makes it available to your RISCO. So I think it's a very simple concept that I'm explaining, and I'm sure there are some real difficulties here because we are talking about in situ populations and all those challenges that go with it. So reaching towards the end, these are the points, 9 questions that I would like to discuss. I know probably difficult to discuss them all here. I don't know if should I?
Thank you, Joana, for your. I like it to hear myself. For your comprehensive presentation, and thank you again for all the speakers of this session. And we now have about 10 minutes left for discussion. And I suggest that we start with points raised by Joana.
Thank you. Okay. I hope you are very interactive today. So I'm not going through order, but well, we can do it. So my first question is, who will actually manage this kind of data, in situ data, and and the in situ material? But who do you think that should be managing this data? Gene banks, protected areas, not farmers, perhaps, but what do you think?
Maybe I can start to answer. While I walk to Nigel, it depends on the data you want to manage. The pure management data of for this in situ reserves must be managed by the reserves themselves. If you're talking about data to access the material, to make users aware of what is available, this could be managed in a system like Eurisco. So it depends.
I think that's what they call the chairman upstaging the question. That's exactly what I was going to say, is it does depend on the kind of data. And I don't think the onus is on the gene bank or the genetic resource center to manage the detailed curatorial data related to in situ populations. But I do think they need then to, one of the questions, and I'm sure it's further down on the list, is whether or not we need to include that kind of curatorial data inside Eurisco, or whether we have some other parallel system for storing in situ data. If we do, then it would have to go via the national focal point, and the national focal point is likely to be located in the gene bank. So there would need to be a relationship between the data management for ex situ and the data management for in situ. But I think as the protected area manager, if we assume the conservation is going to be done in the protected area, then they will already have their data recording system for the species. So we don't want to reinvent the wheel. So we use their system, but perhaps we can look at the system that's used and make sure it's recording the data that we need as people looking at genetic resources, as opposed to their just biodiversity conservation needs.
I could give you my answer, but maybe someone else would like to start first.
Possibly you know my answer already because we wrote a paper on it. In the ideas that were developed in an earlier attempt to create a feasible flow of this type of data into Eurisco, the idea is that each country somehow takes responsibility for inventorying the in situ crop wild relative resources. That's what we focused it on. Extending it to on farm material is a difficult one, so I don't think it would fit, but anyway, it could be a starting point. Having that national inventory similar to Eurisco ex situ material uploaded to Eurisco leaves the management data, and it's quite clear that that flow about in situ resources to Eurisco is aimed at providing access, somehow making it possible for the user to know what material exists in the world. I always give the example of the asparagus breeder in Japan not knowing that on a Dutch island there are wild asparagus varieties which might be very important, or wild asparagus populations which might be very important for the breeding in Japan. Concerning the management, that depends on the type of management you foresee. If you have the reserves that you were talking about this morning, Nigel, I guess that comes with a manager, right? In the Dutch concept, where we more rely on nature conservation organizations, it could be within those organizations or a thin layer on top of those organizations to do a superficial monitoring, safety backupping, and so forth, which is a viable concept because we're doing it at the moment in my country, and that works. So for the crop wild relative, I think we've got a concept. For the on farm material, I'm more worried on how to do that. I think we'll simply lose it if we don't get it into gene banks.
Thank you. Shall we go on?
And do you think that Green Global then could be used for storing in situ data? Adapted and perhaps used.
Wojtek wants to give an answer.
I think it is it very depends on the national conditions. If the documentation system allows to include all data, in situ data and on farm data, it is for national coordinator, or generally for national coordination, it is very, very useful. But if the system doesn't, let's say, accommodate the data, in this case, it can be separately, and well, I think most of us agree that it is necessary to mirror the data into Eurisco. So Green Global or national mutations of Green can allow this, and we started to include the data into Green. And one more point I would like to mention: we have crop wild relatives, wild harvested plant, wild food plants, and so on. There are many categories of this material. It is also good to include material which is generally potential use for anything for humans. So So not only crop alternatives, and also we shouldn't avoid also wild plants that are potential for enlarging species spectrum of cultivated plants. Yeah, so it is a very diverse category.
Thank you, Wojtek. Maybe one last question from your catalog, and then we should focus on the other presentations.
One more, let me see. Well,
so did I understand it correctly that you are planning to put in a fair, accessible database data about the in situ populations of wild crop relatives, make them accessible to everybody?
so the idea is to create such a system that can actually is able to hold that information. Yes.
Yes, and will that be accessible to everybody?
Yes. Well, through Eurisco, yes.
And I mean, wild crop relative populations are sometimes endangered and so on. So don't you foresee any problem with that publicity of the data?
As far as I'm concerned, coordinates is not compulsory to put in the data. So you can actually just describe the population, but not exactly where the species occur. So you don't have someone going there and taking all the specimens. You could point. In fact, in Portugal, we have been talking about it. We could perhaps, rather than giving the right coordinates, the right location, we could point to. We have a database of wild plant species, and many of them are actually threatened because they are in the red book, red data list. So our idea is to point. So this information from the population is coming from that database, and we could actually make I don't know the name in English, but maybe make a link to that database, which doesn't explicitly say where the species are located. So as far as I know, coordinates are not compulsory to put in the system.
That's sort of an alleviation of the problem, but what about long-term conservation of these endangered species? Is there provision for that? Do gene banks have any role to play?
Well, one thing that is different is managing those populations, and that, as Nigel and Theo were saying, that might be done via the genetic resource of the protected areas. And that information obviously needs to be accurate, because if they want to manage the population, then you have to have the information regarding the population. But what then goes into the users is a totally different thing. You don't need to provide any specific detail about that population. You can provide, each country can decide exactly what they want to share, I think. And so these are different things. Monitoring and management of the population won't be available to users. They don't need that information, I think, but something to discuss.
Yeah, the national inventory should be as complete as possible. What is shared via Eurisco is decided by the manager of that database. That's the basis. Referring to the threatened species, we do see in several countries in Europe, and possibly started with the Millennium Seed Bank, that species are being backed up, also non-crop species, ex situ, dried seed, -20, so that they don't die, in case something happens and the species disappears, to create possibility for reintroduction. That's outside of the scope, I think, of the plant genetic resources discussion, but we do see a role of gene banks because of their ability to treat seeds properly in those projects. So, but that's not about plant genetic resources in general.
Okay, thank you. We should focus on the other two presentations. Have you got questions with regard to the presentation of Clara or Catherine? Any comments on this? Then I will start. You mentioned that Clara, recommend to use myopy, to implement myopy for adding phenotypic data to the European information system. Do you think this is feasible to really reach full myopy compliance, to convince the gene banks to provide all the detailed information necessary?
Yeah, I think that that is possible to convince that the gene banks. Of course, I know that there are some people that are still resistant to do this work in our labs, and when I try to explain them the myopy, all people say, okay, this is a lot of work to change all data, but I think that they cover most of the information that they can have for phenotypic data. So I think it is possible to convince them.
Okay, thank you. Are there any comments on this? Any other? Yeah, Giovanni?
So I mean, genetic data, for sequence data, there is a go-to data. There are 2 or 3 go-to databases that everybody uses. What about phenotypic data? Is there something similar to NCBI, let's say, or to EMB?
No, there are some project database like the Harmstone database or some others, but it's not common. That there are not common database to store phenotypic data. Sometimes, for example, you can link or some annotations that is a relationship, but not the data itself. You can find data, phenotypic data for the accessions you are working with. This is not common. In the banks there are, but usually you should go to the gene banks or in the USDA you can find this, but in Europe it's not common to find this. In Eurisco you can find, but only for a few studies. And
yeah, in different gene banks, yeah. You have phenotypic data, for example, in the IPK, in the CGN, in the USDA, but they follow different systems. They present the phenotypic data in different ways. Also, the ABRDC work best in the Gene Bank of Taiwan.
Is there a recommendation from your side how this can be handled in the future? Should it be on Eurisco only, or should there be a dedicated repository for phenotypic information?
Okay, Eurisco has some infrastructure, so it could be a good repository. One of the things could be one of issues is about ontologies, because ontologies are, most of them are specific, at least for the traits, are very specific for a species or organism. So that this general database should change from organism to organism to a specific trait ontologies. For the rest of ontologies, I think that is not a problem, because for describing an experiment of a factor, it's common for all the species, but at least for the ontologies could be an issue. But anyway, I think that could be easy. Eurisco has potential to adapt myopy and standardize the observation and the traits.
Okay, thank you. As we are running out of time, unfortunately, we have to close this session now.